Hi, my name's Dr. Christopher Ward from the Kansas University Medical Center, and today I want to talk about immunofluorescence protocol for detecting polycystin 1. Now, polycystin 1 and fibrocystin are interesting proteins in that they have large ectodomains. Now, we have excellent antibodies for both these proteins, but they bind in the ectodomains. So for polycystin 1 we have 7E12, and for fibrocystin we have an antibody called antibody 11. Now, in the old days we had difficulty getting these antibodies to work. The staining intensity on cells was weak. But on Western analysis we could see the proteins quite easily. So this was a bit of a conundrum. And so we came to the conclusion that perhaps the ectodomains of these proteins could be being swept off the surface of the cells by the fixation process, and in particularly the Triton treatment, which is used to permeabilize cells. And so we have modified our original protocol quite extensively, and the big trick is basically to decrease the amount of Triton that you're using. So we like to grow cells on number 1.5 glass cover slips in a 6-well tissue culture plate, and these can be coated with gelatin and or collagen type 4. Now, the reason we use 1.5 glass cover slips is that we're interested instead in super resolution microscopy, and the STED machine that we have is calibrated to cover slips that are 1.5 in thickness. We get our cells from Professor Darren Wallace's PKD cell repository, and Gail Reef is the lady that actually cultures them for us. And so basically we collect cells from the PKD repository, and they're usually confluent unless otherwise stated. We remove the medium by aspiration or suction and replace with 2 mL of 4% paraformaldehyde and PBS. Then incubate at room temperature for 15 minutes exactly. Then we quench the fixation reaction with PBS containing 100 millimolar ammonium chloride. Now, the ammonium ions react quickly with the aldehyde and inactivate it. You basically get a Schiff base being formed. And the incubation period is 5 minutes, and we do this twice. Then the critical point, we permeabilize the cells with PBS and 0.01% Triton X100 for exactly 10 minutes. Then we remove the permeabilization solution and replace with blocking medium, which is phosphate buffered saline with 10% normal goat serum. And we leave this on for at least 1 hour at room temperature, or we can leave it overnight if you so wish. Now, the idea here is that the albumin in the normal goat serum will bind all the Triton and stop the permeabilization reaction. And also the albumin and the sticky proteins in the normal goat serum will actually then block the cells as well. So then we replace the blocking solution with PBS, normal 10% normal goat serum, and 1/500 of your antibody, if that's 7E12 or antibody 11 or antibody 18 for fibrocystin. And then we incubate overnight at 4 degrees. Then we wash 3 times with 2 mL of PBS. And we replace the PBS with blocking solution with 1/500 of Alexa coupled secondary anti-isotype specific antibody. We get ours from Southern Biotech. Then again we incubate in the dark overnight at 4 degrees, and then wash 3 times in PBS with 2 mL of PBS. And then if you're going to use DAPI, which is a nuclear stain, it binds to DNA, we use it at 200 nanograms per mil for 10 minutes. Now, DAPI is deprecated instead, it kind of interferes with it. But once you've done that, we wash twice with PBS, and then we mount using prolonged diamond anti-fade mounting. So where do we get our reagents? So the phosphate buffered saline is easy. It's PBS tablets from Fisher. The 16% paraformaldehyde that we dilute down to 4%, basically we take 10 mils of this and add it to 30 mils of PBS. And that's from Electron Microscopy Sciences. The ammonium chloride is from Sigma. The Triton is Triton X100, and we get that from Sigma. And we like to get small bottles of it because we do believe it oxidizes with time. And so we get the 100 ml bottles. All secondary antibodies are Alexa coupled and are from Southern Biotech, and we use secondaries that are coupled with Alexa 488, 568, which is of the red color, and the infrared or near infrared is Alexa 647. If we're going to use DAPI, we get that from Sigma. Now you've got to be very careful with the DAPI. It is a DNA dye, and you're putting it at a concentration of 5 mg a mil into DMSO. So please wear gloves. The DMSO will actually very efficiently take that DAPI across your skin if you're not careful. So gloves are a prerequisite to handling it. And then finally we use prolonged diamond anti-fade mountain from Thermo Fisher. So that's a skeleton of what we do, and now we're going to go to the lab, and I'm going to show you exactly how we do it. And then at the very end we've got some interesting confocal and STED pictures to show you, and we'll have a little addendum describing these. So thank you, and let's go to the lab. Hi, here we have some cells from Darren Wallace's core at KUMC, and they're from a normal kidney. In fact, this is normal human kidney 419. And so what we've done is we've grown them on type 4 collagen here, and we have grown them on glass cover slips here. And these cells actually grew very, very quickly. So what I did was I treated them. These have no treatment. These have been treated with 2 nanomolar hepatocyte growth factor, and these have been treated with 2 micromolar heparin and 2 nanomolar hepatocyte growth factor. Now for the type 4 collagens, we've got 4 wells here. So we have a control, hepatocyte growth factor, 2 nanomolar, heparin, and then we have hepatocyte growth factor and heparin. Okay, so we've got all 3 combinations on these cells that are growing on the type 4 collagen. Now they grew very quickly and made a monolayer, and so what I did at 11 o'clock was I came in and injured the monolayer by striking across it with a tip. I did this 3 times, and then allowed them to regenerate until almost 5 o'clock here. So they've had about 6 hours. And when you look at them, the controls are beginning to close the wound, but the hepatocyte growth factor, heparin, are really racing ahead. And so what we want to do here is we want to fix these cells. So what we're going to do is we're going to suck off the media with this suction system here. It's got a container back here for the supernatant. And then we're going to fix in 4% paraformaldehyde. So stop. What I'm going to do here is going to make up a 4% paraformaldehyde, and this is the way I do it. I take a falcon, give it 30 mils of PBS, just by eye really, like that. 30 mils approximately, and then paraformaldehyde. This is 16% paraformaldehyde from Electron Microscopy Sciences. Okay, here you go, there's a box. Okay. So what we're going to do is just So here we need to get a tissue, and we just take off the top one, like that, just break it. And then I just tip it in to make it up to 40. And so we're diluting at 1 and 4, so that gives you a nice 4% PFA. So PFA, or paraformaldehyde, gives a very nice open fixation, kills the cells instantly, and then cross-links all those lysines and free amino groups in a kind of very gentle and open fashion. And the great thing about it is it's not particularly fluorescent. If you did the same thing with glutaraldehyde or formaldehyde, the background fluorescence would be very high. Right, so what we've got is we've got the 4% PFA here, and we're just going to suck off or aspirate the supinate from the glass slides, just like this, very quickly. And then just make sure it's absolutely empty. That's one lot. And I do the little guys, aspirate. Now quickly and gently add your 4% of paraformaldehyde. About a ml each. Doesn't matter if you overdo it, as long as they're immersed in paraformaldehyde. So the cells are killed instantly by this and fixed in position. So all those lysines and free amino groups will be gently cross-linked, and we're not going to incubate them too long in this. Oops, overshot a little bit there. We'll just keep going. And notice I'm wearing gloves. You don't want to get aldehydes on your skin. They're carcinogenic, and they can also kind of cause an allergy as well. That was a big problem back when we were studying anatomy. Everybody got contact dermatitis from formaldehyde, really. So there we go. Beautiful. So very gentle. Just top them up a bit more. So they're fixing right now. Now then, what we need to do is we need to start an accurate 15 minutes. Okay? And we'll do that with a watch here. So we're going, okay? Now one thing I just noticed about these guys is the side, on the side, you notice I put stripes across the edge of the plate. This is in case we mix up the lids. There's important data on the lid, and so this kind of keeps the lid with the bottom, the top with the bottom. And that's a kind of important little thing. It's quite easy to mix them up, okay? Especially if you're kind of busy and you're maybe doing mini preps and trying to do this at the same time. This is Sunday, so we're kind of a bit laid back, and I've got my helper Andrew here, and I'm going to do the same for these guys, like this. Yeah. And a different pattern for the next one. And a different pattern for the next one. Okay, and so give them 15 minutes, and then what we're going to do is we're going to quench them in PBS, which is phosphate buffered saline, okay, which is this stuff, but which has also got ammonium chloride in it. And the idea behind the ammonium chloride is to quench the residual paraformaldehyde. It'll form a shift base almost instantly, and just inactivate the fixation process. You don't want to overfix, you don't want to underfix, okay? And then once we've done this, we're going to permeabilize the cells, and we're going to do that with a very gentle Triton permeabilization step. And we're not going to give it more than 10 minutes with the Triton. Okay, and that will basically make little holes in the membrane so that your antibody can get in. So if we just summarize what we've done to the cells, first of all, with the paraformaldehyde, we've killed them, and then we fix most of the proteins together via free lysines and free amino groups. Okay, but we do it in a kind of open way, so the fixation is very open. Then we stop the fixation, and then we quench it with ammonium to stop that completely, and then we permeabilize it, so there's holes. And then the step after that is then to block it, and we block with 10% goat serum. The 10% goat serum will kind of act as a sort of sticky blocking protein mixture that will basically take out any sticky sites in the cells and kind of block them so antibodies don't non-specifically stick to them. And then after that, we'll go on with our antibodies. Okay, fine. So we're coming up to the end of this paraformaldehyde incubation. What we're going to do is we're going to remove the paraformaldehyde, and we're going to use 100 millimolar ammonium chloride in PBS, and we are going to quench the reaction. Okay, and then leave it for 5 minutes. So we just do this. Want it to be quite quick, you don't want them to dry out. Okay, and then okay. Put that to one side, and then we are going to use the ammonium chloride to just gently on top of each one, just enough to submerge them. And this will get rid of the paraformaldehyde. It will become a shift base, if you remember your chemistry, and it will inactivate it and stop the cross-linking, which is the most important thing. So we'll just keep doing this, and we're going to give it 5 minutes, and then we're going to then use the Triton to permeabilize it after 5 minutes. And that will be exactly 10 minutes in Triton, and I'll explain that in a little bit. You just drop it. Try to avoid the. Ooh, that was a bit rough there. Okay, you see there's an example of what you shouldn't do. Okay, so just. Yeah, we might have blasted off some cilia there, unfortunately, if there's cilia on these. So we're hoping there's some cilia, and we're hoping there's some cells that are healing the tears that we, again, I made a mistake there again. So, and here we go. There we go, that's better. Okay, we'll pop the rest in here. Done. And put that to the side. Put the tops on, and then we're going to give them exactly 5 minutes. So we want to keep things as consistent as possible. 5 minutes. Okay, and that's that. Okay. So while we're waiting, we're going to make up normal goat serum, and it's going to be 10% in phosphate buffered serine. And here we have normal goat serum. It's just from goat blood that's been allowed to clot, and then you spin out the red cells and you're left with the straw coloured serum here. We use it about 10%, and the way I do it is I try not to use too much plasticware. We've got to be ecologically sound these days, and this is very rough and ready. It's a blocking step, and so we don't have to be too careful about things. So I bring up to about just under 30 mils, yeah, just under 30 mils of PBS, and then back on. And then I'm going to add 3 mils roughly of normal goat serum, or NGS as it's called. So this is a very cheap blocking agent. You can go down to the slaughterhouse, and if they're doing goats that day, they'll give you a big bucket of blood, and you can take some of that and allow it to clot, and you can get the serum, and it'll keep you going for a long, long time. So it's just a morning's work to do that. You just ask the guys, so they're just gonna dump it anyway. In Scotland, they'd probably make it into haggis or black pudding or something, but they just dispose of it in this country, so it's a bit of a waste. It's great stuff, haggis, black pudding. Okay, that's it. So we've got a minute to go in the ammonium chloride here, but what I want to point out here is we're going to be using phosphate buffered saline with 0.01% Triton X100, okay? And if you just shake that, it's kind of soapy water, really. I use a very small amount of Triton. I just want to make holes in the membrane, but some of the proteins that we're working with have ectodomains that can be washed off, for example polycystin 1 and perhaps fibrocystin, and so we want to be judicious with the amount of Triton that we're using. Try to use as little as possible, but just enough to kind of open up the cells. And so I've set over the years I've settled on 0.1%. So we've got 20 seconds to go, so let's get ready. And so we just take off we're going to take off the ammonium chloride, and then we're going to add some of this, okay? And then we'll give it exactly 10 minutes, okay? Right, so I'm sucking now. I'll just do this, stop that. So we just aspirate. So paraformaldehyde it's done its job and it's been neutralized, we hope. And you can see the cover slips at the bottom here. And here we've got slide chambers. This whole top finger is going to come right off, and we'll have access to the cells that are growing on the glass here. So here we go. And again. So again, just using the same pipette, we're going to grab some PBS Triton, and again gentle, maybe quite gentle. So it's just like soapy suds. It's a detergent, and just enough to open the membrane but not wash the polycystin off. I think this is where a lot of people are going wrong, especially with 7012, which is looking right at the end terminus of the polycystin ectodomain. You want to be judicious with how much detergent you use. You need some to get it in there, but and then just shoop, shoop, shoop. A little bit in each one. Just wet it. Done. So there we have it. We're going to give that 10 minutes now. Okay, so that's right. 10 minutes is so 10 minutes exactly. Okay, and then it has to be exact. And then we terminate this by adding the normal goat serum. Normal goat serum will absorb some of that Triton, okay, and kind of take it out of the system. And then in the normal goat serum, we'll give it half an hour. But during that half hour, we'll get our antibodies out, and we'll set up the primaries that we're going to use. So we're going to try to do a four colors, okay? So polycystin will always be red, and phalloidin will always be blue. Phalloidin is a stain for actin, for F-actin in fact. And then there are some other proteins that we want to look at. We have two new potential polycystin interactors. One we'll call C16 and the other we'll call CU. And so they will either be green or in the infrared. And the other thing that I want to look at is I want to look at two focal adhesion proteins. One is called talin and the other one is alpha-actinin. And so those two will either go in the green channel or the infrared channel. So we'll set those up in a little bit. Okay, thank you. And so what we're going to do is just take the tritone off. This is, I think, one of the critical things. And if you really want to see what's on cilia and focal adhesions and philopodia, you've got to be quite kind of gentle at this point and quite rigorous in your protocol. That is exactly 10 minutes. Just for consistency's sake. So try to be as quick as you can. Right, so we're doing good. Now then, let's have a look at these guys. They're looking good. Turn that off. Now we want normal goat serum. Just use the same old. And then a ml in each. And this is where you can relax a little bit. You could leave it overnight at 4 degrees if you wanted. That's okay. I like to leave it just 30 minutes and put an antibody on overnight. Well, in fact, a cocktail of antibodies, and we'll see that in a little bit. And just, again, try to be gentle if you can. That's tricky here. Just want to cover them. So the normal goat serum kind of blocks areas that will be sticky for antibodies, and also your secondary antibody is raised in the goat, and so that's the other reason for using goat serum here. Seems to be a lot easier on these little chamber slides. They just fill up quite nicely. There you go. Okay, almost there. Were you coming tomorrow? Good, we're done. Alright, fine. Excellent. Let's make sure everything's is covered. That's good. See these guys are covered with it. And then make sure you got the right one with the right one. Oh, I always double check, you know, it's good practice. And then we're done. And now we need 30 minutes. So let's get time for 30 minutes. Exactly. Ah, that's it. Okay, what we are interested in is we're interested in this new antibody that we've made. It's to a new potential ligand for polycystin 1 called C16, and we're interested in how it behaves. So we're interested in whether it is with polycystin 1 on the focal adhesion. So we have a talin antibody here from Santa Cruz, C9, IgG free. We have an alpha-actinin antibody, okay, which is a rabbit monoclonal. We have another antibody to another potential ligand called 2D11, and this is to CU62, okay, another monoclonal that we've made. And finally, the linchpin monoclonal, which is 7E12. This was made by Albert Ong, myself, and Peter Harris's lab in Oxford, and it's probably the best reagent for polycystin 1 that's available. And what we're seeing is that in cells that are migrating across glass, this appears to be in the polycystin 1 that appears to be expressed in the focal adhesions and in philopodia, okay? And it's co-localizing with definitely with CU62 and definitely with C16, but we're not sure which structures it's on, hence the need for the alpha-actinin and the talon, both of which are markers for focal adhesions. So that's what we're going for. So since this is the most important one, we're going to actually have 3 tubes with our monoclonal in them, and we're going to dilute those 1 in 4 into normal goat serum. Okay. Now we do this because we haven't quite purified this monoclonal yet on a protein A column, but we're working on it. But we're doing some preliminary work at the moment just to see if it's good or not. I just dropped my lid. So we're going to put 2.5 in here. We'll make up 10 mils of each. 2.5. Right here, like that. Fine. I'll just put this to the side for a minute. I'll put a big top on this because we just lost the top there and just fell off. Okay, let's just do that. Right to the side. So that's good. So we've got our new monoclonal there. Change tip. Now we're going to add 7.5 of the normal goat serum. Here it is here. Each one of these. There we go, 7.5. That's 7.5. Doesn't have to be exact with this. 7.5 again. Right, so we've got one antibody sorted out. Okay, there's one other experiment I'd like to do, and that's to look at Cu62 in alpha-actinin, because it too seems to be on the focal adhesion, and it looks like some of the older literature about polycystin being on the focal adhesion, it might be right, especially if you grow the cells on type 4 collagen. And so we're gonna have a really proper look at that as well. So that's one antibody done, so we can put that to the side. Now everybody is going to get 7012 because it's a linchpin antibody here, right here. And so what I'm going to do is I'm going to give everybody 20 microliters of 7012. We'll use this pipette here. Stop it down to 20. Everybody gets polycystin 1, 7012. One, 2, 3, 4. It's a good job I make this antibody because we go through an awful lot of it. So that's the most recent batch. So all 4 of them got polycystin 1. Okay. Now we want alpha-actinin in this first one here. So we'll do that right now, and we'll use it at 1 in 500 again. So 20 microliters. Alpha-actinin. Okay. So there's alpha-actinin. Top of that. And mix. Next one up is going to be talon. Okay. Again, we'll just use it. It's an IgG free. We'll use it 1 in 500 again. There we go. Talon. Double check it. Nice. Monoclonal IgG free. That's good. Right. And now my homegrown antibody against Cu62. Okay. 2D11. And now this one needs alpha-actinin and CU62. So let's bring these up. CU62. Done. Alpha-actinin. And we are done. So we're ready to go. And my timer just went off, so we can now go and apply these to the cells. Okay, so we've given them 30 minutes to block, and what we're going to do now is just going to carefully write. We're going to treat across the plate like this with the different antibodies. So the first one we're going to use is alpha-actinin. Now of course that's in with polycystin and with our new C16 antibody. Then we're going to go talin across this way, again with polycystin 1 on our new antibody. And then we're going to go CU62 with polycystin 1 and our new antibody, see if they co-localize. So the CU62 antibody is actually an IgA, so and the anti-C16 is an Ig2A. Okay, so really kind of different antibodies, and the talin antibody is an Ig3, and of course 7012 is an IgG1. So there's going to be no cross-reactivity between them. And then the very final one is going to be CU62 alpha-actinin. Yeah, and then I'm going to look at these ones on the type 4 collagen again, and I'm going to go alpha A, alpha-actinin. There's not much room here. And T is for talin. I'll write it here, talin. I'm going to write alpha-act. Okay, and then here I'm going to go CU62. Okay. Rightio, and so there you have it. So what we're going to do is aspirate and then put these guys on top, and then we're going to wrap them up and leave them till tomorrow morning. Okay, so let's get on and do that now. So first up, we'll do this plate here, and we'll do these two here. Okay. Okay. Here. Okay, put this here. Now, if you look here, I think you can see where I've disrupted the monolayer, these stripes here. So, hopefully that's where the business occurs, and we get some understanding as to what's going on. So, for this, I'm going to use a 1 ml pipette. I'll just take it real easy. So, alpha-actinin first. Just give it a couple of inversions, just to make sure it's all good and mixed. And one for you. 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 And then I've got a little bit left here, so I'm actually going to feed these, put a little bit more in here in each one of these to make sure they're submerged, so I don't waste any. I think a mil is enough overnight, but I'm being a bit generous here because I can. The alpha-actinin and talin antibodies are a little bit expensive, but as my old mentor Donald Angus used to say, what price is ignorance? And he was absolutely right. So if we do find that polycystin really is co-localizing by super resolution. With polycystin 1, it confirms what's in the literature, but it's a big step forward because we'd be pretty sure that that's right. Okay, now I'm just going to top these up again. Right, fine. So here's tallow. So put the sticky on for tallow. And if you look at the side of the plate, just check that you got the right lid on. Tallow, okay. And alpha actin, just take the sticker off and stick it on there. It's always good practice so you really know what's going on. Okay. And of course you've got it in your lab book, and that's most important. Now we're going to do the next one. Let's put the lids back on here. Okay, CU62 first. So we'll just go with the CU62, see what we get. So CU62 is an interesting protein. It's only found in vertebrates. Whereas C16, it's more widely it's found in a lot of other clades, even insects and things like that. So two quite different proteins. So CU62. That's one done. And then alpha actin CU62. Just to see what happens. We had these, and so we'll have a quick look. So we can be quite generous with this as well, because we don't have a collagen one for it. So these are primary cells from a normal kidney. So that's it. So alpha actin and C62 like that. Now then, one of the things that we have to do is we have to seal these with saran wrap, so we'll just do that right now. And I happen to have some saran wrap right here. Some pattern to them. Yeah, and just stop this. Okay, so now we're going to incubate these overnight. So we really want to kind of seal them as much as we can, so that there's no loss of liquid overnight. It's 4 degrees, and sometimes you see the liquid level go down. You don't want it to dry out. That's the most important thing. And so I'm just going to seal around these like this. Just very, very crudely, but just to stop any evaporation, you know. There we go. I always do it this way. There's one. And 2. So if you don't make your own antibodies, and you buy them commercially, you could actually scale this down. Instead of using a mil, you could go down to maybe half a mil. Personally, I don't like doing that. I think we should do the experiment properly and it's relatively high concentration of the antibody and a high volume as well. Just works better. And you've got to also balance it up against your time. You want an experiment that works. So again, I'm going to just take some of this. I'm going to see around these guys. So first CU. So it's got CU, polycystin 1, and our mystery protein, C16. And the bottom one has got CU62 and alpha-actinin. And then this one here. We've got alpha-actinin, polycystin 1, and C16. And we've got talin, C16, and polycystin 1. That's the question. Do these guys co-localize, or are they in proximity these days with super resolution? So we'll just line these up like this, and we will get them in the morning and put the secondaries on them, and we should be alright. Right, so let's take this to the cold room and we're done. So what we're going to do today is we're going to make up the secondaries for the primaries we did last night. So bearing in mind that everything has got 1712 in it, we're going to look at 1712 in the red channel with a 568 Alexa anti-IgG1. I'm also going to make the actin blue using fluoridin, fluoro at 405. Okay, so we can actually add these to our stock and then aliquot into our individual more specialized antibodies. So it just saves a little bit of time. Okay, we're going to use everything at 1 in 500 just for the time being. So first IgG1. It's a pretty color, isn't it? Something like this. We don't need that again. And the fluoridin. So the fluoridin is a small molecule from Amanita fluoides, destroying the angel or fool's mushroom. Excuse me a second, could you just stop? So now we've got fluoridin here, and we're just going to add that 50 microliters. So then each of these gets 6 mils. So this is just PBS normal book serum, 10%, and it's got the IgG1, it's got a fluoridin in it. So we've got both the actin stain and the 1712 stain in here, and then we're going to adjust the other 2 channels so that we can see the other proteins. So I'll take you through that in a second. So let's just loosely put the tops back on these, and then we'll get on and do this. So each of these is going to get 12 of the other fluorescent antibodies. So we'll start with the alpha-actinin. You look at the lab book, To do the alpha-actinin, we need a rabbit boric gate, and we need an anti-IG2A in the infrared 647. Microliters each. So that's an IgG3, which will be green. And again, we're going to go into the infrared with the IG2A to see our C16. Using an IgG3, quite a rare monoclonal isotype. Good. So that's a talon. Just work through them. So the next one is going to be CU62. And then stop for a second. CU62. So we're going to make the IgA CU62 in the infrared with a 647. Use an IG2A for the C16, and it's going to be in the green channel, so that's like 488. And finally, we're going to go alpha-actinin and CU62. So CU62 is going to be in the infrared channel again, 647. And we're going to have a rabbit antibody as a green. So that will be a 488. It's good to have your lab book open so you know what you're doing. It's probably the trickiest part of it, just getting the secondaries right. With that, we should be fine. So we've made up the the secondaries. You want to keep them kind of in the dark if you can. And put these away, and then we're going to wash down our cells and apply these. Okay. So all of these have had overnight in their primary antibody. And so what I'm going to do is just wash them 4 times, and then put the secondary on. And then the secondaries, it's interesting. It's interesting that kind of leaked here. We'll put the secondaries on for about a couple of hours in the dark, and then we'll wash them and then mount them up. So I'm not using a nuclear stain here, I'm using Floydin to outline cells and to look at philopodia and to look at the focal adhesions. Here we go. This is just PBS. I'll have to gently wash these guys. We're going to do it 3 times. Quickly. Gently. The idea is to get rid of all the primary antibody that you can, and then without disrupting the cells too much. So physics experiment, we're trying to work out whether hepatocyte growth factor has any effect on polycystin expression. I think it might, mainly because the receptor, the c-Met receptor, has IPTG tick domains, and they're also present in fibrocystin. So there might be some sort of interaction going on there, perhaps. Not sure for sure. So, just wash. Get the last bit as well. almost there. So I'd like to give it one more wash. Getting there. I hate it when that happens. So now we're going to put the second drain body on. It's a short kind of wash and incubation. We're going to go for the alpha actinin and the talon. Okay, so the ones that we're going to try to co-localize with polycystin 1 and our novel protein C16. There it is. Get these on. And you're looking at the lid as well to make sure that you don't make any mistakes. Right, so let's go alpha actin first. Alpha actin is part of the vocal adhesion complex. The all I really know about it. So, look at the quick mix. So we're going to do 1 ml for each of these. Just like that. And then I want to go half a ml for the ones that are on the collagen 4. Save a little bit of region here. Just a little color, which is great. Alright, alpha actin. Then we'll go with talon. Double check everything, you know. It's good to be a little bit neurotic, but not too neurotic that you can't do the experiment, so it's a bit of a balance. I find there's a lot of neurosis in science. For some balancing. Don't worry. really want to be sure that I'm right here. There's a literature on the subject, polycystin adhesion. Literature sadly been forgotten about, but I think it might be right. So we're going to go CU62. CU62. CU62 in infrared. Or mystery protein. C16. It's fun making the monoclonals, but it's this is where it gets really tricky, where you're trying to work out where they bind. I find this a lot more difficult. Final one, it can be quite generous. I'm going to go CU62 off acting. And I think the CU62 might actually be on the focal adhesion as well. Which, you know, if we're right, we're introducing 2 new proteins to the focal adhesion. It's meant to have about 200 proteins, so, you know, another couple of proteins will probably not make too much of a difference. Might be quite specialized renal things. Okay. Got them all swapped in secondary antibody now. So now we just need to put them in the dark and kind of forget about them for, well, up to say about 12 hours. Anywhere between about an hour and 12 hours. So I like to just put them in a drawer and come back to them. Okay. Now we're going to wash these. I've had the secondary on for about 4 hours, and I'm just going to wash them all. Top one's alpha-actinin, bottom one is talin. And wash them a few times. I'll wash them just 3 times, quite quickly really. before washing. Okay, so this is for wash.
Yep.
Wash them, get the secondary off them. Right. So what we want to do now is fish out the cover slips, and I use a pair of forceps. And these are the best. It's sometimes tricky to get the cover slips out, but I used to be the best. There's one. Sometimes it's very tricky to get them out. Two. On the side. And then three. Just like just a few drops of this Vector Shield hard set. Put them on top. Manipulate these so they're close together. This one's actually spoke a little bit, you see. A little bit. So what I'm going to just add a little bit more to this one. This. There's two done. A few bubbles here and there, but that's okay. Round it. That's good. Hold that up like that, and then we can get on with the next. Just take that tissue. Next two. Keep them in order. now what we're going to do is we're going to put them away just in the dark for the time being. Overnight, actually, I like to do. And that's that now. Now I'm also going to show you how to do one of these slides. They're much quicker. So you have them. A little bit of a quick What we're going to do is quickly wash these. And the important thing is we want to take the superstructure off here and be left with just the slide. So your chamber slide slots in like this, and your slide remover like this. Just slip it down the side like this. Right away there. Once it's in, and then you just flip it up like this. Gently pull it up. Okay. Right, so we've got a bit of a mess here. We're going to have to pull this out. Grab the glass slide. Just pull this off. Like that. Although this is under off on it, and just examine it, make sure that there's no adhesive still sticking there, and there isn't. Right, and now what I do is I do the other way around. I add plenty of this in the mounting region. Let's fill up the wells if you can. Then I grab a cover slip, pop it off the top like this, as nicely as I can. You're going to get a few bubbles, but we're going to try to squeeze them out rather than do that. Once it's done, it's done. And again, you want to put it in the dark overnight. Okay. Cool. Done. Right, finished. here are some of the cells that I promised you. These have been stained with my staining protocol that I just delineated. And what you have here is you've got two panels, basically the same view. The blue is actually phalloidin, okay, and the green is talin, which is focal adhesion protein, and the red is actually polycystin 1. So let's just take the blue off. Now, what I want to show you is that the red and the green co-localize. So they're kind of making a kind of yellowish color at the moment, as you can see. If we take the green off, you can see this is just polycystin 1 in the red. Put the green back on, which is talin, you can see they co-localize. So let's take the red off, and there's a talin focal adhesions. And then kind of nested within that is the polycystin 1. Let's do that a few times. There you go. So this is with 100 times objective confocal microscopy using Alexa 488 for the talin, 568 for the polycystin 1, and the phalloidin's actually in the DAPI channel right here. And so just put the phalloidin back in. You can see the actins everywhere there. So with that, I'll end my talk, and if you've got any questions or queries, please just write them in the write them in the comments, and I'll get back to you. And with that, I'll end my talk. Oh, and if you like, give me a like. It kind of helps a little bit. So thank you.